Hola, buenos días a todos. Good morning. Buenas tardes, sorry, it's afternoon. Good afternoon to everybody. It is a pleasure for me to have on this occasion of our seminars, Tiana Kohlsdorf. I met her when we were working at Gunther Wagner's lab. We overlapped for a very brief period. I think she was actually visiting in this process of leaving of leaving the lab. But we had interesting conversations, and I've always been fascinated by her research, and some of the nicest stuff I've seen about reversion, for example, come from her studies of limb reversion in limbless lizards, which is the topic that we're going to talk today, but also because she has this very wide view, okay, that includes behavior, that includes EvoDevo, and I was just talking about with her reaction norms, right? Phenotypic plasticity, and this is really hot stuff that we think is the path to follow in having a complete understanding of the evolutionary process. So it is my pleasure to leave you with Tiana. Thank you, Tiana.
Thanks, Alex. Thanks a lot for introducing me. Thanks a lot for the invitation. I must say it's an honor, a big honor to be part of the seminar series. So just share my screen here. I think that will, yeah. Is that working? Can you all see the screen now? Yeah? Okay, good. So yeah, as I said, I'm really enthusiastic about being here because I'm really enthusiastic about talking about science, and really enthusiastic with EvoDevo and EvoDevo in general. Today I decided to bring a seminar that focuses more on the evolution of snake-like body shapes in tetrapods, because I think in this seminar I can combine results from different studies that have been contributing for a better understanding of those multiple origins of snake-like phenotypes or morphotypes in different lineages of tetrapods. I always like to start my EvoDevo talks or EvoDevo talks, bringing this chart from the Field Museum in Chicago, because I tend to say that's basically impossible to not be puzzled or astonished by the huge amount of endless forms of that are really beautiful and diverse in our planet. And a question that often comes to our minds when we see such diversity is to ask ourselves how biological diversity evolves at all. So basically, this is a core concept or a central question in the field of EvoDevo, the short name for evolutionary development. Some authors prefer to use Devo-Evo from developmental evolution. I think Gunther Wagner is one of those researchers that prefer to use Devo-Evo recently. And basically, this is a field that integrates evolution genetics and developmental biology to address questions related, for example, to the origin of new features or evolution of general patterns in specific lineages. So just to recapitulate or recover a little bit what's the idea of looking at development to understand evolution now, or biological evolution. I will just bring some examples here that help us to connect those two dimensions, development and evolution. This is a chart for proposed for a hypothetical population. Can you see the mouse here, the cursor? Yes? If I move here.
Yes, yes, we can see.
Right. So let's think about this hypothetical population here. If we take a look on the distribution of phenotypes or phenotypic variants in the population, let's say, for example, body size, we expect quite often, not always, but quite often a distribution that would be like that. So in this hypothetical population of frogs, for example, some individuals are really large, some are really small, and most individuals are near what we would call the phenotypic average. This range here of phenotypic variance correspond to the revealed variation, which is actually the variation that's selectable, or what would be seen in some way by natural selection, for example. So what happens is that those revealed phenotypes, they are actually revealed by development. So development integrates genotypic information with environmental signals. Those could be general environmental signals from developmental environments like temperature or UV radiation, but could also relate to cellular, the cellular development, for example, in the embryo. And development integrates those signals in the process of revealing the phenotype. So if we go back to the hypothetical population that I brought before, developmental mechanisms, they can actually directly affect the amount of variation that's accessible to selection. Let's say that some change could be a mutation, could be some change in the developmental environment of those animals, could be a combination between those two, like for example, developmental processes becoming more sensitive to environmental signals. But let's say that some change in developmental processes result in some frogs that are really, really tiny in this hypothetical population. In this situation, development would actually increase the range of revealed variation that's selected. And then if this new variants, novel variants that are revealed by development in this new condition or from this new mutation, they are for some reason favored in this condition, then we can expect a shift. So the shift would be in the phenotypic average. So after several generations, what we could say is that the phenotypic average could be in a new position in this graph. So just to synthesize, selection can shift the phenotypic averages, but it does not originate new phenotypes, right? New phenotypes are revealed or are originated through changes in development. And that's why it's so interesting to look at development and evolution. The relationship between or among all those dimensions in evolutionary processes, these relationships have been expressed by this diagram from Oster and Albert. And basically they connect information contained in the genotypes that interact with developmental environments, expressing cellular and molecular networks that actually reveal possible phenotypes. And those possible phenotypes can be then either selected or change along generations through drift. And that's when we see what we would call the established patterns, which are the phenotypes that characterize, for example, a given lineage or a given population. Although EvoDevo more often concentrates studies in this part of this diagram, looking at the origin of phenotypic variation, this is part, those dimensions, they are actually part of a continuum. And in our lab, we're interested in taking a look on all those dimensions and connecting all of them. But today I will focus more on this part of the diagram, and I will be concentrating most of the studies that I will bring along this line, right? So I will not approach the interaction between genotypes and developmental environments, although we are really interested in that. And these relationships are really interesting to discuss. So in order to address some of those connections between those different dimensions, we have been using quite often the genotype-phenotype or GP maps approach, which is an approach that consists of searching for molecular signatures in the genotypic space and trying to establish connections between those molecular signatures at the genotypic space and the phenotypic patterns that we identify in the phenotypic space. And the logic behind this connection here is related to the idea that the developmental space is what connects the genotypic space with the phenotypic dimension. So we've been looking with more attention at genes that are expressed during developmental processes. Although we use more often the approach of gene candidates, so we look at gene candidates and choose some specific regions of the genome to look at, we recognize that one gene more often is involved in the development of several traits, and a given phenotypic trait is actually the result of a combination of different genes being expressed and interact to each other. Okay, so let's shift now to the main morphological pattern or phenotypic pattern that I will address today. I will focus on snake-like body shapes. And if I ask anyone, like I ask my kids to think of a snake, probably this is the overall shape that will come up to your minds. So this is the famous snake-like morphotype. It's a morphotype that evolved independently in several different lineages. So these are some of the classical and beautiful snakes, but you can also see this snake-like body shape characterizing amphibians, some legless or almost legless lizards like those microtates here and here are also characterized by snake-like morphotypes. And we can find snake-like morphotypes even in lineages that are external to amniotes, like, for example, the Sicilians here that belong to Gymnophiona. So as I mentioned, the snake-like body shape evolved independently several times in different lineages. Squamates have been by far the most studied group in terms of those recurrent origins of snake-like body shapes, because almost all big lineages within squamates have representatives of the snake-like body shape. But we can also see, as I mentioned, the snake-like body shape evolving in this amphibian. So what characterizes the snake-like body shape? I bring here three main features, which are the absence, or at least limbs being vestigial. So limbs are absent or vestigial. Sometimes animals that have the snake-like body shape, they lack autopodia, sometimes which are hands and feet. Sometimes they lack limbs, sometimes they have no girdles also. The other feature that characterizes a snake-like body shape is the large number of vertebrae. It's amazing the amount of vertebrae that those animals have. And some authors claim that the trunk vertebrae, they lack identity. This is a general expression to refer to a morphotype, a lumbar morphotype that has ribs associated to the vertebrae in this region. So what I will do now is to focus on those three main features that characterize the snake-like morphotype. And I will just bring some examples on how we have been applying this approach of genotype-phenotype maps to add some insights from EvoDevo, from an EvoDevo approach to the discussion on how this pattern that characterizes the snake-like lineages, how it evolved in different groups or specific groups. Okay, so let me start by the first feature that I mentioned, which is the characteristic of limbs being absent or at least vestigial in some of those lineages that evolved snake-like, the snake-like morphotype. As I mentioned here, there are several lineages that evolved a snake-like morphotype, and I will focus here on knowledge that we have from EvoDevo studies using representatives of Gymnophthalmidae, which is, are the microtates, and from snakes. So current literature already points out what will be or would be the main developmental pathway that we should focus if we are interested to understand what are the developmental mechanisms that changed during the evolution of limbless species. So this is a really useful figure that comes from this work here by Liao and Co. back in 2016. And what we see here is that there are basically two main pathways that would be like really good candidates to take a look on if you're interested on limblessness, or at least evolution of extremely reduced or vestigial limbs. If we start by looking at a general lizard, what happens during the development of during limb development in a general lizard, such as and also in mice and other animals that have pentadactyl limbs, for example, is that there is a region here where a gene called sonic hedgehog is expressed, and the sonic hedgehog pathway is a key pathway during limb development, and expression of sonic hedgehog in limbs is modulated by a limb-specific enhancer, which is a ZRS, and combining patterns, expression patterns of sonic hedgehog with Hox expression, then the limb bud grows and also differentiates, right? What we know from snakes, also available in the literature, is that there is a huge change in this limb-specific enhancer, right? They have a big deletion, and that affects the whole pathway of sonic hedgehog expression during limb growth, or at least in the origin of the small limb buds, affecting the whole process of limb growth and differentiation. Especially if you think about hind limbs, that some snake species, they have vestigial hind limbs in some specific stages of development. Okay, so I'll just focus first on ZRS, which is this enhancer here, right, that modulates expression of sonic hedgehog. This is what we know from literature. So this is the deletion that snakes have in this enhancer. And what has been interpreted in literature is that this deletion here explains why snakes don't develop also the vestigial hind limbs that show up at some point of development. This is the big deletion. In this recent paper that just came out from a group that looked at convergent loss of limbs in reptiles, looking at the whole genome, they interpret that the changes in ZRS, they explain divergence based on a given element that is lineage specific. This huge deletion was only identified by them in snakes. But if you take a look on other different elements near some specific genes and pathways, then it's interesting that they can find some shared divergence. So I've been looking at the ZRS, not looking at completely limbless species, but looking at patterns, different patterns of limb reduction, focusing on one specific group of lizards, which are the gymnophthalmids. So those are the microtates. If some of you are interested on lizards that have a neotropical distribution. So this is a really interesting family because it's a family where we can identify two clades that experienced limb reduction and body elongation and evolved the snake-like body shape. So one lineage is this one here, which is called Gymnophthalmini, and this is the other lineage which involved Bacchia lizards. So those are some of the snake-like lizards that we can find among those microtates. Those are Calyptomatus and Notobacchia. And hopefully you can see here, they have really, really tiny hind limbs. So those are extremely small. And they are, in some of those species, they are like a single digit stiliform hind limb. And they lack in this lineage, they entirely lack the front limbs, right? So we have addressed, as I mentioned, we are interested in the whole, in the connection of all of those dimensions that are involved in evolutionary processes. So we know some of the implications, functional implications of this specific body shape in terms of association with specific habitats and microhabitats, relationships with climate, and some effects in terms of burrowing performance in those lizards. But today I will focus more on the morphological patterns that we see in those different processes of digit loss and limb reduction in Gymnophthalmini. So as I mentioned, in this family, 2 different lineages independently evolved morphotypes that are limb reduced. One thing that's really interesting about this group is that while in Gymnophthalmini, limb reduction was more concentrated on the front limbs, many species entirely lack front limbs. They keep some vestigial hind limbs, sometimes with just a single digit, like you can see here. In contrast with Bachia lizards, the pattern is the opposite. So in this lineage, limb reduction was more concentrated in the hind limbs, and several species retain front limbs, sometimes with having 1 or 2 digits. These are some examples of the really weird reduced morphologies that you can see in these lizards. This is, for example, a hind limb of a Calyptomatus with just a single digit here. This is the extremely reduced pattern we can see in Bachia. And one thing that's really interesting, we have characterized the muscle anatomy of those reduced limbs in Gymnophthalmids in collaboration with Virginia Abdala from Argentina. And we see a huge reorganization of not only bones, but also muscles in those lizards. And if you take a look on this combination of bones and muscles in those extremely reduced limbs, some of the morphological patterns, they actually resemble what studies doing genetic manipulations in mouse, for example, found. So, for example, this is a mice mutant for sonic hedgehog. If you mutate sonic hedgehog in mice, you get a hand that has a single digit that really looks like, not externally, but internally in terms of bones and muscles, to what you see, for example, in Scriptosaura or Calyptomatus, right? And the same if you play around with other aspects of the sonic hedgehog pathway, you'll find sometimes polydactyl limbs that have digits all having the same number of phalanges. We already have some information also from literature about sonic hedgehog expression, at least in one of those lineages that experienced digit reduction. I must say it's almost impossible to get embryos from these embryos, but these lineages, but Juliana Rossito, which was a student in a different lab here at USP, she managed to get some of those embryos. And what she found was that sonic hedgehog is actually expressed during the development of those really tiny vestigial hind limbs in Calyptomatus. But expression is really transient, and they interpret that the failure in the maintenance of sonic hedgehog could explain this stelliform autopody. Okay, so this is focusing on Calyptomatus and this single digit hind limbs, but Bachia lizards also have a really interesting story in terms of digit evolution. In this group, we have statistical support for reversals of digit loss, and this is a feature that has been striking me for over, I think, the past 15 years, I think. Not only we have statistical support for reversals, and I'll focus on those 2 species here, Bachia scolecoides and Bachia panoplia, but the pattern of those reversed, those autopodia that reversed digit loss is a really unique pattern. So if you compare here the 2 species that apparently reversed digit loss, and they have 4 digits, they are tetradactyl, but all digits, they have exactly the same number of phalanges, which is patterns that resembles what you see, for example, in mouse that have the GLI3 knockout mutations, right? So this is a pattern that's different from the ancestral condition for lizards in general, and it's also different from the ancestral condition inferred for Bacchia lizards. In terms of musculature, the muscles of those limbs that reversed loss are really different also. So this is the hind limb, or the feet of the foot of a Bacchia scolecoides, which apparently reversed to the condition of 4 digits. And if you take a look on muscle anatomy, the muscles really resemble a hand of another species, a more ancestral species that has a higher number of digits. And we have dissected also Bacchia panoplia, which is here. And in terms of musculature, they have the same pattern of Bacchia scolecoides, which means in terms of muscle configuration, the feet look like hands, right? In terms of osteology, they have this really weird osteology where all digits have the same number of phalanges, both in pes and manus. So in the Bacchia re-evolved autopodia, the configuration of digit 1 in all digits having 2 phalanges and the unique muscle identity really calls our attention, and makes us think or question ourselves if this reversed tetradactyl state is similar or homologous to what was the ancestral tetradactyl state in this group. So what I did more recently was I decided to sequence the sonic hedgehog enhancer in game of Talmini. So this is the CRS, which is that region that in snakes, it comprises a huge deletion. And after sequencing sonic hedgehog, both in the game of Talmini lineage, which is this one here that has these animals with those really unique stiliform hind limbs, and also in Bacchia, including the Bacchia scolecoides, which is the one that reversed digit loss. These are the patterns that I found. Basically, most of the mutations or differences in this enhancer are concentrated in the lineage that has a stiliform autopodia. And we can actually identify some sort of degeneration of this enhancer in this lineage. This degeneration apparently is a degeneration that has some functional implications. So if you make after making a motif estimation, what I found is that motif 7, which is this one, is lacking actually in this lineage. This is the lineage that has the animals with the stiliform hind limbs, but it's present in Bacchia, right? So Bacchia, in Bacchia, the motif 7 is conserved. And this is a motif that apparently comprises a transcription factor binding site for hawks. So apparently the whole hawks sonic hedgehog pathway is modified in Calyptomarus, but not in Bacchia. Not in Bacchia that reversed loss like Bacchia scolecoides, or Bacchia that have extremely reduced autopodia, lacking several digits like Bacchia flavescens. So the hypothesis that we came out from this study is that maybe reversibility of digit loss in Bacchia lizards is explained by this combination here of continuity of developmental information despite of the intermittent character expression in the group, right? But remembering that, or keeping in mind that the re-evolved digits, they are morphologically really, really different from the ancestral condition, right? So they keep this history, let's say like that, in the expressed phenotypes. On the other hand, digit loss may be irreversible in Gymnophthalmini because the enhancer degenerated in this group. Okay, so now I'll just shift to the second feature that I mentioned, which is the large number of vertebrae observed in snakes, amphibians, and other animals that have the snake-like body shape. So those are two X-rays just to convince yourselves, if you're not already convinced, that those animals have a huge number of vertebrae along their trunks. Going back to that chart that showed a little bit about what are the main developmental pathways involved in limb loss. Probably remember that HOX is also a pathway that's involved in digit development and limb development. But this pathway is also involved in body elongation. So we have from literature information that HOX-A13 in particular is expressed in the tail buds of animals, different animals and different lineages in tetrapods. When HOX-A13 is expressed, usually this protein, this transcription factor, will decelerate the process of body elongation and also decelerate the process of mitogenesis, right? So there is a lot of information available in the literature showing or suggesting how HOX genes control the vertebrate body elongation. And basically it's an effect of HOX, for example, HOX-A13 in the wind pathway mainly, which also involves FGF pathways and notch segmentation, right? So this has been already described in a general perspective in mouse, and even comparing with expression patterns observed in snakes. So how can we apply the GP maps that I mentioned, genotype phenotype maps that I mentioned as a key approach to this context? What I've done back a long time ago was just first take a look on the sequences of HOX-A13, focusing on the first exon of HOX-A13 in snakes and also amphibians and Sicilians. And we can identify a signal for positive selection in the HOX-A13 exon 1 in snakes. And this signal for positive selection is not found, at least in this region of the genome, in amphibian and Sicilians, right? So the signal for positive selection comes from mutations in 5 amino acid sites. Those are the 5 amino acid sites, just to illustrate here in the alignment. So they're spread along the alignment. And what we've done more recently was to take a look on what could be some of the functional effects of these snake HOX-A13 signatures in the context of body elongation, right? Can you see me? Alex, can you confirm that's all working here?
Yes, I can see.
Okay, right. Okay, perfect. It's just that it was frozen here, but okay, I'll just keep going. So what we did was, after identifying those 5 sites evolving under positive selection in snakes, we decided to snakeify HOX-A13. So we used an approach where we combined expression vectors of having the mouse HOX-A13 sequence, and we have a comparison with the same vector having the mouse HOX-A13, but this vector was snakified. So in the 5 amino acid sites where we identified positive selection, we replaced the mouse amino acid by the snake one. And then what we did was we transfected HEX cells using either the empty expression vector containing the mouse sequencing and the sequence, and the expression vector having the snakified HOX-A13. And we looked at the effects both using luciferase assays. Those are focusing on specific physical interactions between the transcription factor and specific enhancers, and also using a genomic approach using transcriptomes based on RNA-seq. So in terms of transcription factor physical interactions, what we observe is that the mutations in the snake-fied hoxatrotin, they actually affect the physical interactions of this transcription factor. So you can see here with the red bars. Also, the snakification of hoxatrotin also affected the expression levels in different pathways in general. So by comparing results that we obtained using either the MT expression vector or the mouse, the vector containing the mouse sequencing with the snake-fied hoxatrotin, we found more than 1,000 genes differentially expressed. Most of those genes, they are related to processes involving anterior-posterior pattern formation or some mitogenesis, which are upregulated in this condition. And this is interesting because those are the patterns that, the pathways, sorry, that usually are repressed in the presence of hoxatrotin in mouse, for example, and that stop body elongation and somatogenesis. But snakes not only are really, really long and have a really long, a huge amount of vertebra, they also have what some authors call the trunk vertebra that lack identity. So this is the third feature that I will approach now. If you take a look here on the top, I have the example of a skeleton of a lizard. And here in this region, you can see this is the lumbar region of a lizard that has vertebrae that lack those really long ribs that characterize this thoracic region. So if we compare this lizard with a snake or with a Sicilian at Gymnophiona, in both cases, you see that the lumbar region is not morphologically recognizable based on the presence or absence of ribs. So this is a morphotype that we call rib-associated lumbar morphotype. And part of the developmental pathways involved in the process of growing up ribs in specific places of the trunk, they have been already characterized. So basically the establishment of a difference between a lumbar region and a thoracic region involves this transcription factor regulatory element unit here. We call HOX10 H1MIF5 transcription factor regulatory element unit. I know this is a huge name with lots of letters, but the idea here is if we think of an embryo developing from a lizard in general that has the lizard-like body shape and has a clear lumbar region, what happens is in the region that will originate the lumbar region, when HOX10 is expressed, it interacts with this regulatory element here, which is called H1. And this is a regulatory element that will repress expression of MIF5. So in this region where MIF5 is not expressed, then the lumbar region doesn't develop thoracic, doesn't develop ribs, sorry. Current literature suggests that polymorphism in this region, this regulatory element H1 in snakes actually blocks interaction of HOX10 with this region here, and that in turn won't repress expression of MIF5. And that's how we could explain that in one specific region where HOX10 is expressed, the ribs remain being formed and grow despite of HOX10 expression, right? So what we did was we started to map this polymorphism in this regulatory element in different lineages of tetrapods. So the polymorphism has been previously described in snakes and also in Afrotheria. So elephants and manatees, for example, also have this polymorphism in the regulatory element. Recently, we have identified that it's also present in Sicilians, and we sequenced a whole bunch of other snake species and also confirmed the presence of this polymorphism. What's interesting is that there are some snakes that actually lack this polymorphism. So apparently they reversed to the ancestral condition that would allow interaction between hogstem and this regulatory element. But in this species that reversed the polymorphism, they have some specific mutations in Hox genes. So apparently the interaction between those two parts of the interaction unit, the transcription factor and the regulatory element, is maintained. The lack of this interaction is maintained in these lineages despite of the reversal. So what we conclude from this third group of results is that this rib-associated lumbar morphotype can be or probably evolved in different lineages associated with changes in either one of those components of the transcription factor, regulatory element interaction unit. So either a change in the transcription factor or the regulatory element, or both, will result in a similar phenotypic outcome, which is having ribs associated to a region that would correspond in other animals to lumbar region. So just to conclude and bring some of those general insights that EvoDevo provides to studies focusing on the multiple origins of snake-like body shapes. The first feature that I've approached is the fact that these animals are characterized by limbs being absent or vestigial, and information from the limb-specific enhancer of sonic hedgehog, which is ZRS, can help us to understand not only how some of those animals have those really tiny vestigial single-digit hind limbs, but also add some information in the discussion of reversibility of digit loss in specific lineages. In terms of the large number of vertebrae, some insights from HOX-A13 pathways and the molecular signatures that can be present in identified in specific coding regions can contribute for our understanding on the increased number of vertebrae in those animals, and the rib-associated lumbar morphotype can be investigated using this approach that combines transcription factor with regulatory element in these interaction units, suggesting that changes in different parts of the developmental pathways can result in similar phenotypic outcomes. Okay, so I think in general, those are the main messages that I decided to bring today to this seminar. This is the group. In general, we are trying to move more frequently to lab meetings in the lab instead of Zoom or Google Meet, but this is a nice picture from the pandemics. I want to just emphasize the presence of Aniele. So Aniele is the person that did all the study on the third topic that I brought today on the transcription factor regulatory element interaction unit. Marina is a former student from the lab, and she did all the HOX-A13 study using transfection of cell lines and so on. These are the funding. We keep fighting to keep our funding strong in Brazil, despite different movements trying to take out some of the funding that we have in the country, but this is essential for doing this research. And yeah, and this is our website if you want to take a look on other studies or other things that we've been doing, for example, in reaction norms, developmental plasticity, and also working with other types of animals. Okay, so I think that's it. We can probably open for discussion and questions. Thanks a lot.
Thank you very much, Tiana. Very impressive, very impressive. We have questions. Does anyone want to go before I go?
I would like to ask something.
Sure.
Tiana, do you see this correlation between limblessness and elongation of the body and thoracic-like vertebra, the relation between, why do they evolve together usually? Can we go maybe beyond genetic regulation? Maybe there is a causal sequence in those processes. Let's say you elongate the body, somehow you can't develop, you don't have a big enough lateral mesoderm to develop limbs, to say something. Do you see any kind of maybe stronger developmental causality of this correlation?
Yeah, I think there is, unfortunately, fields, I'm talking about biology, keep not talking to each other that much. So there's a lot of, a huge amount of studies that actually address body elongation with digit reduction or limb loss from a functional perspective. So many of those snake-like body shapes, they evolve associated with specific microhabitats or specific habitats. So for example, if we think about fossorial species, fossoriality would be, seems to be enhanced, not the fossoriality, but the performance, locomotor performance under the surface seems to be favored by long bodies that actually lack limbs, so they are like head first burrowers. And because those animals adopted completely different locomotor style, the changes in both directions in terms of body elongation and limb reduction together could improve or enhance locomotion in those specific settings. The same would happen with grass swimmers. So if we think about grass swimmers, some long lizards are grass swimmers, and when they just move, slide down along the grass, for example, limbs would be most of the time together with the trunk, so that they can, that would decrease friction. And that could also contribute for performance. So there is a huge group of information focusing on biomechanics, locomotion, that addresses the concomitant, those two processes being sort of concomitant in terms of body elongation, limb reduction. Then if you take a look on what comes from genetics and developmental biology, I think most of what we have is actually associated to or comes from snakes. I think that the fewest genomes, complete genomes that came from other snake-like body shapes came in the past 5 years. But from snakes, there is a hypothesis that I will call elongation, let's say like that, of hog's expression along the body would actually, this is a change actually in terms of the expansion of the field, expression field of hog's genes along the body, actually results in disappearance of the field that marks the front limb, where the front limbs will develop actually. So for snakes, at least disappearance of front limbs could be explained by this pattern of an expansion of the expression field of hog's genes along the trunk. That could be associated also with elongation. We only focus now on HOX813, which is the gene that's playing a role in terminating the process of somatogenesis and body elongation, but it's not the gene that modulates the initiation of this process. So if you take a look on HOX genes that initiate or coordinate the initial processes of trunk elongation and somatogenesis, expansion of the field, expression field of some of those genes could result in the disappearance of the marks along the trunk that would initiate the budding, limb budding in the front limbs, but not in the hind limbs. So that is also one thing that could explain why some snakes retain vestigial elements in the pelvic girdles and so on. So I think that if those two fields start to talk to each other, probably what will happen is that we can see a combination of functional implications of elongated trunks that are limbless in terms of biomechanics, especially in specific contexts like fossility or animals that are grass swimmers, while some of the developmental processes can explain how those features change together, right? Because that's what you see. Usually the third feature that I brought, which is related to the rib-associated lumbar morphotype, this is something that's not general for all snake-like body shapes. Some animals have two of the three features of the snake-like body shape. They are long, have lots of vertebrae, they lack limbs or have vestigial limbs, but they have some specific region that has at least reduced ribs, if not absent, that could be interpreted as a lumbar region or sort of that.
Thank you. Obrigado.
Welcome. I forgot to mention that if anyone wants to come with questions in Spanish or Portuguese, it's okay. Spanish, despacito. I will not answer in Spanish because my Spanish is terrible, so that's why I'm keeping in English. But it's not a problem if you want to discuss in Spanish with me answering in English.
Yeah. So I would like, just to make sure I understood well, you found that the ZRS enhancer is still functional in some of your limbless lizards. Maybe it's got something weird about it, but it's mostly there. And among those, you have some that have actually recovered their limbs, right?
Yeah, sort of.
Right, yeah. Kind of recover. While in those that have more extensively altered their ZRS enhancer, you have 0 cases of reversion. That's it. Okay.
Yeah, one thing that I want to point out, maybe I went too fast, and certainly I forgot to mention that, is that if we take a look on gymnophthalmids, those microtates that I'm interested on, so far there is no species that's completely limbless in terms of front limbs and hind limbs. And this is really important information because the program for building up digits, for example, was not completely erased, because you can find in both lineages species that retain either, even if it's a vestigial hind limb, but it's a vestigial hind limbs with phalanges. And the same with Bachia. So with Bachia, most the digit loss is concentrated in the hind limbs, but there is no entirely limbless Bachia species, right? So the program for building up digits is still there. What happens is that in one lineage, apparently digit loss is irreversible, and this is the one that has the enhancer that degenerated. And in the other one, we have evidence, at least from phylogeny, that digit loss could be reversed, although the reversed morphology is not exactly the same in terms of morphology and everything, anatomy. It's not the same of what you see in the ancestral, what would represent an ancestral morphology in this group. And this is the one that has the enhancer conserved.
That's interesting. I'm just thinking about this because we had this idea that neutral changes change the reaction norms or the epigenetic field, if you wish, of possibilities. So I think that would be perhaps an example, in the sense that in some of these lizards, they were limbless. From this limbless condition, your ZRS may continue to degenerate without perhaps a significant impact on the phenotype. You're still not limbless, sorry, almost limbless, digitless. Mm-hmm. You continue to degenerate silently, and that lineage can no longer recover doesn't have that possibility in its morphospace, if you want, while the other lineage, which this change has been silent, right? One of being drifted, more degenerate ZRS, whereas this other is also limbless, but it could still recover the ZRS. So I'm thinking that that could be a nice example.
Yeah, I think that makes totally sense. Yeah.
And I love your recovered limbs, that they're so weird, because these are like zombies, where they come back, but they're not the same, right?
Yeah. Yeah. Well, initially, I was even postulating that actually this is a good example of the prediction that a reversal would imply in a morphotype that has some scars from the time where, because it's not evolving from the same ancestral condition, right? So it's different to think of a tetradactyl, like a 4-digit toe that evolved from a 5-digit ancestral, and then think of a 4-toed limb that evolved from a 2-toed ancestral condition, right? And the scars, they are actually anatomical scars. They are there. So the limbs are different. The 4-toed from a pentadactyl ancestral is different from the 4-toed that evolved from a 2-toed ancestral condition, right?
Kind of reminds me we have this bird, the Pelagornis. It has tooth spikes, but they're actually bone, right? They are developing a very tooth-like pattern. And current thought is that they have co-opted some of the old tooth pathways. So it is a reversion in a way, but there's no enamel, no dentine, all those genes are degenerated in birds, and it acts on osteoblasts instead. So it's the tooth has come back, but it's really improvising with whatever's left there, let's say, and it's another nice example of zombie reversal is what I joke.
Yeah.
Oh.
Yeah, exactly. Yeah.
Okay, do we have more questions? Anybody has another question? Let me see, maybe somebody on YouTube. It's just someone saying hi on YouTube. Sebastian Zapata says hi, in case you know him. Well, okay, that was very interesting. Great talk, and absolutely impressive. The kind of stuff you're doing at all levels, you know, molecular and morphological. It would be really great to have you again so that you can talk about more of your cool research. All right, everybody, I guess it's lunchtime.
Thanks a lot for inviting.
Anybody else has the last opportunity for making a question? Okay. All right, people. Thank you very much, Tiana. Great talk.
Bye-bye. Thanks a lot. Bye-bye.